this is the second half of a lecture on diversity in viruses and bacteria for Bio 101 for August 26, 2020. And it's going to focus specifically on bacteria, in particular what a biofilm is and how it's related to humans, and also talk about what an antibiotic is and how we as humans and patients have created an antibiotic crisis in the United States. And so we organize living organisms into 3 domains. We talked a bit about hierarchy in a previous lecture, and so we're going to look at the structure of domains and kingdoms today, or those levels. And so the domains that life is organized into are basically 2 domains of prokaryotes, or organisms that lack a nucleus and organelles in their cells. And these are the archaea and the bacteria. And then a 3rd domain in which all the organisms do have a nucleus and organelles, or known as eukaryotes. So there are 3 domains of life. There's the archaea, the bacteria, and the eukarya. Archaea and bacteria are prokaryotes because they don't have nuclei or organelles in their cells, and eukaryotes, including fungi, plants, and animals, do have a nuclei and organelles in their cells. And so today we're really just going to focus on these 2 prokaryotic domains, the archaea and the bacteria. And then we'll move on to domain eukarya and talk about fungi, plants, and animals in later lectures. And so the archaea and bacteria are both microscopic, and it's important to note that microbes are everywhere. There are about an estimated 1 trillion species of microbes on the Earth, and over 99% of them have yet to be discovered and characterized. And that's because in order to really study and discover bacteria, you have to be able to study them in a lab. And a lot of them are located in places like the bottom of the ocean in geothermal vents, or the bottom of a geyser that we can't reach, or buried deep within the dirt. And so we can't actually get to them to study them or discover them, right? But there's this huge amount of microbes covering the Earth. And in addition, it's not just the Earth that microbes are covering, it's also the human body. And so there are about 3 microbial cells, or 3 bacterial cells, for every 1 cell in the human body, which means that there are about 100 trillion microbial cells or bacterial cells living on or inside of you at any point in time, making up what's known as the human microbiome, which is all the microbes that live either on or in a human body. And it makes up about 10% of your body weight, just for some reference. And so the first domain of microbes that we're going to talk about are what are known as is domain archaea. And archaea, we know the Latin root, right? Archaea is old. And so these are the old bacteria. They're the original life form, and they came into existence about 3.5 billion years ago, which is a very long time. And at this point in time, the Earth's atmosphere didn't really have oxygen. It was mostly made of carbon dioxide. And so these bacteria were what are known as anaerobic, or without oxygen, so they don't need oxygen to survive. There also was no other life forms for them to use to get energy, right? So it's not like they could eat a plant or something like that. So there had to be a way for these archaea to get the energy they needed to survive. And that is by converting sunlight into chemical energy. And that means that many of them are what's known as phototrophic. The photo standing for light, trophic being for energy, right? And so archaea don't need oxygen. They can take sunlight and turn it into chemical energy. And they tend to live in places that were protected from some of the harsh environment of that early Earth, right? So it was not a nice place to be 3.5 billion years ago. It was extremely hot. The atmosphere was very harsh. And so bacteria, these archaic bacteria, had to find good places to live. And so they still live in those places today, even though the environment has changed. And they're what's known as extremophiles for doing so. They still inhabit what are known as extreme environments, right? Where the temperature is extremely high, or they're really salty, or they're exposed, like in this picture of the geyser, to a high level of sulfur in a geyser. And so we find archaea living in these crazy environments, or these extreme environments, in particular the hydrogeothermal vents at the bottom of the ocean. And so we don't, other than that, there's not really a ton that we are going to talk about in regards to archaea. It's important to know that they're sort of these old bacteria, and they lived successfully in a world without oxygen by inhabiting extreme places. And they still do that today. But when we think about bacteria in general, the ones we think about are the new bacteria, or the ones that are part of domain bacteria rather than domain archaea. And they evolved about 2.3 billion years ago. The first one in domain bacteria is what's known as cyanobacteria, or a blue-green algae. And so you can see a picture of cyanobacteria down here. And the reason cyanobacteria has sort of changed everything is it was able to take the CO2, or the carbon dioxide in the atmosphere, and use that to make food. And as a byproduct, it actually released oxygen into the atmosphere. And that changed everything. So cyanobacteria is the reason why the rest of life likely exists, because we all are aerobic, or use oxygen to get energy. And without cyanobacteria converting that CO2 in the atmosphere and releasing oxygen as a byproduct, we would not be here today. And so that happened about 2 billion years ago. And since then, bacteria have expanded and diversified, and so has the rest of life as well. And so when we think about archaea and bacteria, they are really different at the very level, like the very lowest levels. The sequence of their DNA, for example, is very different between an archaea and a bacteria. But they do share many characteristics, because they're both prokaryotes, right? So they're those types of cells that don't have a nucleus or organelles. And so you can see a typical picture of a bacteria over here on the right-hand side. And what you'll notice is that there's really not a lot going on inside of this cell. There's some DNA, or nucleic acid, right? It's loose and it's stringy because it doesn't have a nucleus to house and organize it. And there's really no other structures inside of a prokaryotic cell. They're relatively simple. And so you see that chromosome or DNA labeled here. And then one other important structure for both archaea and bacteria is what's known as a cell wall. And so there's a membrane inside of the cell, which kind of keeps everything in place. But then there's an extra layer of protection on the outside, which you can see here, and that's known as the cell wall. It's made of a lot of different sugars and proteins, and it helps to protect the archaea and bacteria from the outside environment. And it also gives them a shape. So the cell wall is there to provide some structure and some protection for this cell. Because as we remember, these organisms are living in the environment as a single cell. And so they need that protection around every single one of their cells to keep them safe. And the composition, or what the cell walls are made of, differs between the domain archaea and domain bacteria, but that's sort of beyond the scope of this class. Right, and so originally, scientists thought that the prokaryotic cells, bacteria, archaea, just lived alone in isolation as one single cell in the environment. But we now know that that is not true. Most bacteria live in what's known as a biofilm, which is a community of bacteria or microbes that's held together by this sticky kind of sugary matrix, or kind of like a sticky goo that glues all the bacterial cells together and holds them together as a community. And this matrix, this sticky substance, is secreted out or released from the different types of bacteria, and then it binds them together, and they can actually interact with each other within the biofilm. They can communicate They can pass genes back and forth, and they can start to behave as kind of a group rather than a single cell. And you've probably seen a biofilm before. Here's one on the top of a stagnant bit of water. Here's a biofilm that's actually inside of a lead pipe from, this is a picture from the water systems in Flint, Michigan. There is a very high level of contamination in that water that you may have heard of or read about in the news. One reason for that was chemicals that had leached into the water. The other was bacteria. And the bacteria were actually forming these biofilms, which were really difficult to get rid of and were infecting patients, right? And so biofilms are great for bacteria. They make them stronger. They give them a nice, safe place to live. But this is bad for humans because, one, biofilms are really, really hard to get rid of. That sticky matrix sticks very well onto surfaces. It sticks very well together. And it's hard to deplete, like it's hard to destroy it. And if it's so hard to destroy it, we can't destroy the bacteria that are living in it that might be making us sick. And so, for instance, in a lead pipe, it might be hard to destroy this bacteria, and people drinking that water might become infected as a result. And so biofilms are good for bacteria, unfortunately bad for humans. Biofilms can also form on the surfaces of implants, and so medically they pose a problem because bacterial biofilms can form on something implanted in your body and lead to infection that way as well.